Welcome back, friends. So in this video I am going to continue with the female cone. Now the female cones are not true cones. They are not true cones because the Megasporophylls, they do not form a compact ovulate strobilus, okay, or the female cone. They are not arranged in a compact structure just like in the case of the male cones. Instead, the Megasporophylls, they are loosely arranged in close spirals around the apex of the columnar trunk, and they are situated among the crowns of the vegetative leaves, the scaly leaves and the foliage leaves. We can see here in this picture, these are the Megasporophylls which are situated among the crowns of the foliage leaves, which are situated at the apex of the trunk. Now these Megasporophylls, they are arranged like an ordinary crown of the foliage leaves, and they are collectively termed as the female strobilus. Yes, you can see here clearly in this picture. These are the Megasporophylls. They are arranged just like an ordinary foliage leaves, a crown of foliage leaves, and collectively termed as the female strobilus. Now, in this case, one more difference between the male cone and the female cone is that we have studied that in case of the male cone, they showed sympodial branching. As the apex of the stem, it stopped its growth, has been used in the development of the male cone. As the male cone is formed, the apex of the stem, stop its growth. Okay. Now, whereas in the case of the female strobilus, here the apex of the stem continues its growth by producing a next crown of foliage leaves. That means the female plants, they do not show sympodial branching. As the apex of the stem, it will continue its growth. Okay. So the Megasporophylls also, they arise in acropetal succession. I had mentioned this in case of the male cone, acropetal succession, where the young Megasporophylls are found at the top and the old Megasporophylls are found at the lower portion. Now, the Megasporophylls, they arise in acropetal succession once in a year between the successive whorls of the foliage leaves, and when they fall off, they will leave a persistent leaf base on the stem. So just like the foliage leaf, when they fall, they leave a base on the stem of a plant. Now let us take a look at the Megasporophyll. The Megasporophyll, the crown of the Megasporophylls, they develop only when the plant is quite old. So we will find the Megasporophylls only when the plant is old. Here this is another Megasporophyll of another Cycas plant species. The Megasporophylls, okay, you see here, this is yet another picture of a different type of Cycas, female Cycas plant. Now, if you take a look at a single Megasporophyll, they are approximately 10 to 23 centimeter long, and and they are leaf-like pinnate structure. They are brown in color and they are also covered with hairs. Some Megasporophylls are smooth and some are covered with hairs. You can see here in these other pictures, you can see they are covered with hairs and they are leaflet pinnate structure. Okay, you see here these are the pinnate structure of the Megasporophyll. Now if you take a look at the single Megasporophyll, this is another Megasporophyll of Cycas plant species. So let us take a look at a single Microsporophyll. Now this figure, it shows the different types of the Megasporophylls of Cycas plant species: the Cycas revoluta, Cycas cericinalis, Cycas rhomphai, Cycas bedome, Cycas pectinata, and Cycas siamensis. Now, one thing which is common in all these Megasporophylls is that the upper part of each of the Megasporophyll is pinnately dissected into several segments. Okay, they are pinnately dissected into several segments which may be considered as the leaflets of the pinna. Okay, these are considered as the leaflets. Now each Megasporophyll here, if you take a look at all these Megasporophyll, they usually bears 1 to 5 pairs of big lateral ovules. Okay, you can see some ovules are small and some are very big. Now the Megasporophylls in some Cycas plant species, they are very long and the ovules are many, which attached in pairs. Okay, they are attached in pairs and the ovules are quite in some Cycas plant species the ovules are small, whereas in others they are very big. Now here we can see that in some Cycas plant species, we can see here that they are very much dissected, forming many lobes or pinnae. Okay, forming many lobes or pinnae, whereas in the case of this figure B and C, in case of Cycas cericinalis, we can see that the pinnae are absent. Okay, they are absent. That means they represent there is the presence of only the short spines. We can see here short spines which represent the reduced pinnae. That means the pinnae here is very much reduced, whereas in others we can see clearly these pinnate leaf. Now leaflets. Now the Megasporophylls. Okay, we will study in detail about the structure of this ovule. Now this ovule, the ovule of the Megasporophyll or the Megasporangium. In case of male plant, we call it a microsporangium. Remember, the microsporangium, it carries where the pollen grains or the microspores are formed. So in the female plant, we have the Megasporangium or the ovule. Now each microsporangium or the ovule, they are sessile. Okay, that is they are directly attached to the Megasporophyll without any stalk. They are sessile and they are orthotropous in position. Orthotropous in position here meaning the ovule is straight and upright with the micropyle at the apex. Okay, these type of ovules are called orthotropous. Now, they are ovoid, spherical. They are either ovoid or spherical, and also they vary in their sizes. Where we see here they vary in sizes from species to species, such as in the case of Cycas circinalis, the ovule may attain the length of 60 centimeter. So, a mature ovule, it consists of a massive nucellus. You can see here, this is a massive nucellus, and this massive nucellus is okay, we see here in a mature ovule, we have a massive nucellus surrounded by a thick integument. You can see here, this is the integument and it's very thick. And also you can see here that the integument is differentiated into 3 layers. Okay, the inner layer, the middle stony layer, and the outer fleshy layer. These are the 3 layers of the integument, and it is very, very thick. Now these ovules, they are dark green in color, but they usually turn bright orange or red when become ripe seeds. Now also the surface of the ovules, they may be smooth, as in the case of Cycas circinalis, or they are covered with brown ramenta. Okay, they are covered with brown ramenta, or we call it a brown thin scales which cover the ovule. Now let us take a look at the development of each of the megasporangium or the ovule, how it developed. Now the development of megasporangium in Cycas is eusporangiate type. Okay, it is eusporangiate type, meaning having a sporangia. The sporangia, it arises from a group of epidermal cells. Okay, let's take a look here. This is the Megasporophylls, and how this ovule it developed. It is developing from, it arises from a group of epidermal cells. That is, the development is of eusporangiate type. Now this cells, this ovule, it developed from a group of hypodermal initial cells. These group of initial cells, usually 4 to 6 cells, on the axis of a young Megasporophyll, they will start dividing. Okay, they begin to divide. Around 4 to 6 cells will divide, and these cells will divide and redivide to form a small outgrowth or a protuberance, which gradually increases in size, and it forms what we call the nucellus. Okay, so first we have the initial cells which arise from the hypodermal initial cells. Okay, these group of initial cells, 4 to 6, on the axis of a young Megasporophyll, they will begin to divide and redivide until they form a small outgrowth which gradually increases in size and form the nucellus. The nucellus, that is the central part of the ovule, the central part of the ovule containing the embryo sac. Okay, so this is the central part of the ovule. It starts to form by the division of the hypodermal initial cells. Now at the same time, what we see here, there are some neighboring cells. some new neighboring cells of the nucellus, they become meristematic. Okay, we know that the meristematic cells are actively dividing cells which will give rise to the new tissues. Now here the neighboring cells of the nucellus also become meristematic. They will start to grow and divide around the nucellus. Okay, they will divide around the nucellus and form a ring-like outgrowth called the integument. You can see here in this figure B, this is a ring-like integument. The integument. Okay, so the integument here, what you can also see in figure B as well as figure C, though it continues to divide and grow, but it does not completely cover the nucellus. Instead, it leaves a small opening at the top. You can see here in this figure D as well. It leaves a small opening at the top, and this opening is called a micropyle. Okay, for this micropyle, we know that it is a small opening in the surface of an ovule through which the pollen tube will penetrate, okay, during pollination. Now the pollen tube will penetrate through the micropyle. So here at first the nucellus and this nucellus and the integument, they are free. Okay, they remain free, but later on, due to the growth or due to the intercalary growth, the fusion is not complete. Now the integument, okay, due to intercalary growth, the nucellus and the integument will fuse. But we see here in this case, the fusion is not complete. It is not complete we can see here that the nucellus and the integument remains free at the micropylar end, but they are fuses at the chalazal end. They fuses together. Okay, so the apex of the nucellus projects into a micropylar tube and forms a nucellar beak. You can see here in this figure, it is clearly shown here that the apex of the nucellus will project into a micropylar tube and will form a nucellar beak. Now some of the upper cells of the nucellar beak, they will break down and form a flask-shaped cavity called the pollen chamber. So we have a pollen chamber. What is this pollen chamber? It is the place where the pollen grain will germinate after pollination. So here we see that the integument, okay, in this figure. Also, what do we see here in this? You can see here the integument is forming here in figure B, C and D. Then later on, this integument, it will differentiate into 3 layers as I had mentioned before. These 3 distinct layers, that is the outer fleshy layer, it is also called as the sarcotesta, the middle stony layer, that is the sclerotesta, and the inner fleshy layer, that is the inner sarcotesta. And you can see here these things in black, they are nothing but they are the vascular bundles. Okay, they are the vascular bundles. Now regarding the vascular supply of the ovule, only 2 vascular bundles will enter from the Megasporophyll. Okay, from this Megasporophyll they will enter into the ovules. Only 2 vascular bundles. Remember, there are many vascular bundles in the Megasporophyll. Only 2 they will enter into the Megasporophyll. only 2 vascular strands. Now the vascular strands, the outer strand consisting of a dozen of vascular bundles, enters into an outer fleshy layer. You can see here this one, it will enter through this outer fleshy layer. Even this side you can see here that it sorry, that it enters to the outer fleshy layer, and also they extend up to the micropyle. Okay, they extend up to the micropyle. Now the second strand, you can see here this second strand, they also consisting of many vascular bundles, they traverse the inner fleshy layer in both sides. You can see here these are the vascular bundles which enter up to the inner fleshy layer. Now also one thing you have to remember in case of the vascular bundles, that all the vascular bundles in the Cycas species, they have a massage xylem. Okay, they have a massage xylem. Now what happened next? Okay, in the development of this ovule, what happened next after the nucellus and the integument has formed? What happened next is that one of the deeply situated cell of the nucellus, we can see here many cells inside the nucellus. Now one of the deeply situated cell of the nucellus, it will become larger in size with a prominent nucleus and a dense cytoplasm. This cell, it will get differentiated and it will function as a megaspore mother cell. Okay, I repeat that out of the many cells in the nucellus, one of the deeply situated cell, it will become larger in size with a prominent nucleus and dense cytoplasm, and this cell will get differentiated and it will function as a megaspore mother cell. So here next what happened, this megaspore mother cell, it will elongate and divide by meiosis. Okay, now after the division, it will divide meiotically to give rise to a linear tetrad of 4 megaspores. You can see here clearly in the figure H where we have a linear tetrad of 4 megaspores. So this one, megaspore mother cell, it is shown here in figure E to H, where it divides through meiosis to form a linear tetrad of megaspore mother cells. Now these 4 megaspores, out of these only one spore, okay, remember this, only one spore, that is the lowermost one. The lowermost one meaning the one here which faces the chalazal end. Okay, so the one facing the chalazal towards the chalazal end, it will become as a functional megaspore. Now what happened to these 3 megaspores? These 3 megaspores which lies towards the micropylar end, they will degenerate. Okay, you can see here they starts to degenerate and they will provide nourishment to this developing megaspore cell. Now this functional megaspore, you can see here this one, this functional megaspore is regarded as the first cell of gametophytic generation, and it remains embedded inside the nucellus and develops into the female gametophyte. Okay, so this is how the ovule develop. Now next, in my next video, I am going to explain the development of both the megaspore, okay, that is the female gametophyte, as well as the microspore. Let me just show you, as well as the microspores. Okay, we see here there are so many microspores form inside the microsporangia. So we will study the development of these microspores, or the male gametophyte, and the megaspores, that is the female gametophyte, in my next video.